down. And that's what we thought, and we were wrong. So actually, Sally, if you could take that poll away, and then what we'll do is we'll move on and I'll show you some of the data we've got from this study. Or can I make the poll go away? I can. Look at that. Awesome. Power technology. Okay, so we were interested in how marine invertebrates that live in the tidal flats deal with being in this continuously moving sea of mud. And to test the effects of suspended sediment on the invertebrates, we went out and we collected cores of sediment with the invertebrates intact, and I'll show you what that looks like in just a minute, from 3 separate places, brought them into the lab, exposed them to different amounts of suspended sediment, and then saw how that affected their both taxonomic diversity, which is the species names, and the functional diversity, which is more about their ecosystem function, so where they live and what they eat. We had a high intertidal site from Kingsport, which had relatively few species and a low abundance, a mid intertidal site from Evangeline, and a low intertidal site from Evangeline as well. And this is a site that's about 2 kilometers out from the shoreline. The low intertidal site had the most taxa, the most functional groups, and the highest abundance. And so when we looked at the field cores, we had 3 different field sites that had very different estimates of diversity to begin with, both in terms of taxonomic diversity and also functional diversity. So to do this experiment, you need a lot of undergraduates who have good, strong legs and good, strong backs. And then you head out onto the tidal flights and you get some sediment, bring it back to the lab. And so what we did was we wandered out to our sites and we collected cores of sediment with the invertebrates intact and very carefully transferred them into these plexiglass pots and brought them back to the lab. And so the sediment structure is hugely important to the well-being of these invertebrates. And if you disrupt the sediment structure, they don't do very well, and it can take them a long, long time to get reestablished. So we very carefully took a core of sediments with the animals intact and brought them back to the lab. Packed them up into coolers, and they were quite heavy, dragged them back to the lab, and then grew them, put them in these benches called mesocosm benches, which I will show you in the next slide. So the mesocosms are these large stainless steel bins, if you like, where we can put the cores. So there's a few cores in here, and these plexiglass racks. This was very early on in the experiment, so everything was still very clean. It doesn't take long until everything gets covered with mud. The mesocosm benches that we have at Acadia are super cool because you can run them on ambient tidal cycles, and they'll advance the tide at whatever interval you want. So say 50 minutes on every 24-hour interval. This is very important because these invertebrates, even though they seem very simple, have behaviors that are entrained to the tidal cycle. And so if they are entrained to thinking that low tide is going to be now, but they're in a lab situation and it's high tide, they don't know how to feed. They don't know how to deal with that. So the presence of water can give them some hints that, oh, maybe I should start to feed now, but their behaviors are really entrained to that tidal cycle. So these mesocosms are really great ways of providing a good habitat for the invertebrates. Some of the cores, and you can see our cores with animals in here, we're in a mesocosm with low suspended sediment, so 10 to 40 milligrams of sediment per liter, and that would be what you'd find in surface water in the summer. And others were in this other mesocosm with high suspended sediment, 100 to 400 milligrams of sediment per liter. And again, this is a lot of sediment. And we needed a lot of aeration and pumps to just keep that sediment moving all the time. And so that would be more typical of what you'd find in the sediment water interface, where there's just, so the sediment, the water moving over that, there's a lot of moving sediment. And of course, that's where the benthic invertebrates are living, and they're inundated with this big moving wall of mud. So for the field sites, what we found was that the low site had the greatest diversity, and the site highest in the intertidal had the lowest diversity. These plots show that. These are called NM MDS plots, and basically they're a form of cluster analysis. So each of the dots represents one core, and the data for that one core include all the species and the abundance of those species in that one core. And the cores are 10 centimeters across, and you can have between 300 and 400 invertebrates in that 10 centimeter diameter core. A lot of these are very, very tiny polychaetes, and so you can have very high numbers of them. The cores that are clustered closer together are more similar in terms of their diversity, and the cores that are further apart in these plots have a different diversity. So here you can see the cores from the mid and the high intertidal were very similar in terms of the species that were present, but also in terms of the functional diversity. So who was a burrower, who was actively motile, who's a herbivore, who's a predator. So these cores were very similar in terms of the functional diversity, but the low intertidal site, again, was the most unique. And in the low intertidal site, 57% of the taxa were only found in that low intertidal site. So this is not rocket science. We expect that when you move across a tidal flat, that you'll get an increasing diversity as you move more close to the water line. But it just gives us a good place to start for the rest of the analysis. Okay, here's a little bit more data from that same study. And again, the question is, how does a drop in or a change in suspended sediment influence the animals that live on the tidal flat? These are more NMDS plots. So each dot represents one core, all the animals that lived in one core. Green are field. Red are from the lab treatment that had the high suspended sediment. And blue, the lab treatment that had the low suspended sediment. And the different shades of blue are different time points, and that doesn't matter for us today. So field, high sediment, and then low sediment. These NMDS plots cluster cores by similarity based on functional groups, so not taxonomy, but where they live and what they eat. And what they show is functional groups that were found all the way across the tidal flat. So they were found in the high intertidal, the mid intertidal, and the low intertidal. They were totally fine when the amount of sediment was decreased. So this is a group of species that are burrowing, motile, and don't have jaws. This is a group of species called interface feeders. So they feed in that sediment water interface. When there's low water and they're exposed, they scrabble up particles from the sediment surface. And then when the tides come in, they put their tentacles up and they grab food out of the water. And they were both totally fine, regardless if there was high or low sediment in the water. But the functional groups that were only found in the low intertidal were lost. So they were fine at high sediment, but as soon as we moved to low sediment, they were gone. And so what this tells us is that the low intertidal site that had the greatest taxonomic diversity also has the greatest functional diversity. Some of the groups that were only found in that more specialized habitat were not very able to cope with a rapid drop in the amount of suspended sediment. So does it matter if we have a loss of biodiversity from the low intertidal zone? This is a good question. Biologists are always going to tell you, yes, biodiversity is important because it is. But these two pictures of the tidal flat show you representative images from that low intertidal site. These are footprints of migratory shorebirds. There's a lot of migratory shorebirds, semi-palmated sandpipers and others that come through, and they eat a variety of things, but the small polychaetes and the small amphipods that are found in these sediments are a major source of nutrition. So a loss of diversity of some of those species could have a big effect on the migratory shorebirds. And this image here shows fish bites from Atlantic sturgeon. So this is a fish bite, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, at least 13 different fish bites. And so the sturgeons are coming up on the tide, they take a bite, and then they spit out the sand and they eat the worms. They take a bite and they spit out the sand and eat the worms. And so if there's a loss of diversity, it could have a big impact on some of these species. So biodiversity in a sea of mud. Surprisingly, most invertebrates that we found out there were adapted to a high suspended sediment load. And we didn't look at the delicate sponges and bryozoans, although that would be because would be easily choked out by the sediment, but that would be an interesting thing to follow up on. Change in sediment load had little effect on the species that were really widespread, but a change or a loss of suspended sediment had a big impact on the community that had the highest diversity, both in terms of taxonomy and also in terms of function. So that's just a little bit about biodiversity in the Minas Basin. Sediments are extremely important. But the basin itself, I think of as being quite an extreme environment, in that the sediments are continuously moving. A lot of the animals that live in the upper intertidal zone could be aerially exposed for about a third of a tidal cycle over a month. There's very long periods of exposure. The traditional foods that you think of primary productivity, phytoplankton, aren't there, so they rely on detritus. It's a very, very odd environment. And then since the sediment's moving all the time, there's a lot of erosion and deposition of sediments. And so you might be in a patch that's really very stable over a long period of time, but a sudden storm or change in the way the water is flowing over the tidal flats can really just sort of wash out your whole habitat. So do invertebrates living in this environment have specialized adaptations for life in this environment? And as Tamara mentioned, I'm a developmental biologist. I'm not an ecologist. I'm a developmental biologist. One thing I'm really interested in is developmental plasticity. So developmental plasticity is the capacity for species to change the way they develop in different environments. And it's called eco-devo in some worlds, how development is influenced by the ecology of a species. And the example I want to talk about is a little worm, not surprisingly, called Polydora. And they're super cool in that they can produce three different types of young, or three different morphs of young. They lay their eggs in egg capsules, and some of the young develop into these small larvae that hatch as small swimming larvae, and they can disperse for a couple of weeks. So they're the sort of comes from away. They'll move into a new habitat or new population and become established someplace else. Their full siblings grow really rapidly, and so this is one of the juveniles developing in that same egg capsule. So there can be full siblings. They grow very rapidly and hatch out as juveniles. And the way they do that is by eating the small ones. So they can eat the non-developing nurse eggs, but they also eat the small larvae. And this is a larva here. There's hard to see, but some larval spines sticking out here. And in section, we've actually seen entire larvae that have been slurped right in by one of the juveniles, and just the whole thing just being ingested. So this is SIB cannibalism, and it's very common in this particular species. And so the question is, if I just back up, the question is, we know we have the species that has developmental plasticity. They make small dispersing larvae and large cannibalistic juveniles that would presumably recruit right next to their parents in that same habitat. The question is, what are there environmental cues that can drive larvae to take one particular path, so the path to dispersal, or another path, the path to cannibalism and local recruitment? And we've worked on a variety of different potential environmental cues with, I'll say, not a whole lot of success, but the one that has been working really well for us recently is methionine. So why methionine? Methionine is an essential amino acid, so we have to have it in our diets, and polychaetes do too. It's present in very, very low amounts in salt marsh detritus, and that's the major source of food for these polychaetes and many other invertebrates is the salt marsh detritus. And it's present in really, really low amounts, and therefore it's considered to be limiting in the diets of marine detritivores. Marine predators get lots of methionine, marine herbivores, lots of methionine, but those that rely on detritus have a really hard time getting it in their diet. The other reason methionine is of interest is that it's the universal methyl donor. And so, what does that mean? Methionine donates methyl groups to a lot of biochemical processes, including DNA methylation. So, there's a field of research that is all the buzz. You can read about it in Maclean's magazine and on CBC podcasts. They talk about epigenetics all the time. In epigenetics, what you